Hey guys, this is Kendall Terry, and this lecture is on translation. We're going to specifically start with the tRNA molecule that you can see behind me here, because that's kind of where we'd left off with the other one. We'd made mRNA, and now where do we go from there? So when we look at tRNA, there's two different drawings we can look at it. This is kind of a flattened out model. You can see the 3' end, and it makes these 3 loops and then comes to the 5' end. There's a piece that sticks off right here. This is the acceptor end, and this is where the amino acid will attach. And down here, this loop is the anticodon loop. This will have the code that matches a specific codon of the mRNA molecule. So this molecule won't be flat like this. It'll actually fold up, and it gives an interesting shape similar to this. This is the best rendition I can give you that's still a flat drawing, but it has a little bit of this kind of shape with the anticodon down here, and the acceptor end will be this point up here. This will be the shape that will draw the tRNA from here on out. The T stands for transfer because it transfers the amino acid to the ribosome. When we look at the ribosome is made up of 2 subunits. This bottom one and then the top one here. This top one has 3 sites, the E site, the P site, and the A site. Let's look at each of those sites individually. The P site binds the tRNA attached to the growing peptide chain. And you'll see that in just a minute, but that is the peptide bond will happen between the P site here and the A site amino acid. So it's holding that new grouping of peptide bonds in the P site. The A site bonds to the tRNA carrying the next amino acid to be added. A amino acid, you can see the wording there. This one will have 1 amino acid hanging off the end. This one could have multiple amino acids right here. So this has a bunch of polypeptides, P site. This has an amino acid, A site. The E site bonds the tRNA that carried the previous amino acid. So from the P site, it will shift to the E site, which is the exit, E for exit. And at this point, it will no longer have any amino acids attached to it. Let's see this in a little bit more detail. All right, so here we have the initiation part of translation. All of these that we've talked about, replication, transcription, and now translation, have 3 main steps: the initiation, the elongation, and the termination. So for initiation to take place, we've talked already before that it has to start at an AUG, that's the start codon. It will bind to a tRNA molecule that's carrying methyladenine. There is some difference here between prokaryotes and eukaryotes. Prokaryotes, it's a special methyladenine called formal methyladenine. In eukaryotes, it's just regular methyladenine. So there's a little difference there, not one to stress too much right now. So when this AUG is here on this subunit, notice the other part of it is not here yet. Methyladenine being carried by the tRNA attaches, and at that point, that signals for this larger subunit to attach as well. So now you have this larger subunit with the open E site, and it's got an open A site as well, sitting there ready now for the next step of translation. All right, so then we get to elongation. We're going to start right here in this one. This is a cycle, so it will just continue this until you get to the termination sequence. So you've got the methyladenine is attached to the P site, that tRNA there. So now there's a new code in the A site. Here comes the matching tRNA that has that amino acid, and it's going to sit into that site. You can see that here. So now you have 1 in the A site and 1 in the P site. When both of those are present, there will be a peptide bond formed between those amino acids. At that point, they will shift. What was in the P site will shift to the E site and will exit. What was in the A site will shift to the P site. And now you've got a new A site that's open. That will bring in another tRNA and so on and so forth. As this cycles, it will grow an amino acid chain. Which is the whole point. That amino acid, as it grows, you get a polypeptide, and then eventually that polypeptide will be your protein or bind to some others. Okay, so now we have the termination sequence. In the termination sequence, we have this growing links of amino acid the E site has been emptied, and now the A site is open. When the A site is open, there's a codon there. In this case, let's say that codon is UAA. That's a stop codon. So that will bind to a release factor. It sits in that spot just like the tRNA did, but it does not carry an amino acid with it. So as that sits in that A site, that signals for everything to break apart. So you'll have the small subunit will break, the mRNA will go on and either make another one or be dissolved. The big subunit will go off, the tRNA will come off, and then this amino acid will be released. This polypeptide at this point will be released and then do whatever it does in the cell. All right, so the final thing is mutations. Mutations has been one of those things that we keep coming back to in this whole process. There's two different types of mutations that can affect what we're talking about. Point mutations, which alter a single base, one nucleotide difference. And there are three different subcategories of that. A substitution is where one base that should be there is removed and a different one is put in. So it should be a T, but instead of a T, it's a C. Or it should be a G, but instead of a G, it's an A. So you've got a substitution that's taken place. A nonsense mutation is a codon that was an amino acid sequence, because of a point mutation, is now a stop codon. So now that amino acid chain that you should be building, that polypeptide, gets stopped short, and then you obviously get no protein that gets made. A frame shift mutation is where you have an addition or a deletion of a single base, and everything after that shifts. Remember, codons are read in threes. So if you have this, and then this one gets taken away, what happens is this one will shift over. So you'll get these three that will read together, and now you've got these three over here that will read. So everything shifts. Or if you get the addition of one, that one will be here for these three, and then you'll have three that will have to come out over here from it all. That's kind of hard to show on my hands. I don't have multiples of three, right? So you've got a shift in the frame, and you can think of this as like a sentence. If you take a sentence and you insert a new letter, and everything else would have to shift down a little bit, that's going to cause some interesting words possibly to be developed down the line. A chromosomal mutation is a change in the actual chromosome. So this looks at just one base pair, so a point. You can point to this base right here has been altered. In a chromosomal mutation, you're talking about several nucleotides that are being altered at one time. A deletion, where you have a piece of the chromosome just completely broken off and disappears. We see this in a condition that we've looked at in class, some at least, called Cry-du-Chat, which many times is a lethal mutation, meaning that that individual will have either shortened life expectancy or no life, one or the other. Duplication and adding a copied region. So you have, it should read AAA, or the gene, A gene, and a B gene, and a C gene. And then that should be it, and instead it's another A, B, and a C gene added to that. So it increases the length of the chromosome. Most of the time, this doesn't have any negative side effects unless you're looking at something being overproduced, and if overexpression causes a problem, then that can be very negative as well. And inversion is part of the chromosome is broken in two places, inverted and reattached. Now, that could go on with no problems, but if that broken is in the middle of a gene and then inverted, that gene is no longer going to read correctly, so that could obviously cause problems down the road. And then finally, translocation, where part of a chromosome is broken off and then it's attached at another non-homologous chromosome. So let's say chromosome number 1 breaks off, and part of that part that broke off is attached to chromosome number 4. So now you've got different signals and different expressions and possibly different promoters and that sort of thing. That gene may never get used again because it's over there and it doesn't make sense anymore. Now, although the majority of mutations that take place inside of organisms have no side effect, negative or positive, there are some that do have a negative side effect. Those are the ones, obviously, that we focus on most of the time. But there are occasions where it gives you a positive effect. That's natural selection. Natural selection was driven by mutations in the DNA. And without those mutations, natural selection doesn't take place because there's no variation then. So don't think of mutations as always negative. Most of the time, it's no effect. Some of the time it's negative. Some of the time it's positive and leads to evolution.